So this is the 20 amino acid serine glycine alanine linker here. And this is a GP120 from a CCR5, or an R5 as we call it, virus Val strain. And there's the D1, D2 human or rhesus. Why rhesus monkey? Because when we go into rhesus macaques, we want a homologous system that does not induce antibodies to CD4. So if you use human CD4 and you go into a monkey, you get a lot of reactivity against the CD4 sequences themselves. So we express this full-length single chain. So we refer to this as full-length single chain all the time. We expressed it in 293 human cells, and one other experiment expressed it in CHO cells. Make the protein, and this is with protein alone, not DNA prime boost, not trying to really optimize things yet, but just seeing what would happen with this construct, just protein alone. Well, we did it. Here's the full-length single chain, human full-length single chain rhesus. We originally began with a chemical-linked CD4 GP120, and we left this approach because of the complexity of making one molecule, making another, then the chemistry of linking them together. So it was far too complicated. And although the results were interesting, they were more variable. These are some of the earliest results presented here. Here means France before. And it was strong neutralization of a variety of laboratory strains that are so-called R5 or X4 that use the CXCR4 or CCR5, or that are dual tropic, and the titer. And then this showed that this complex of GP120 and CD4 had much greater breadth of inducing neutralizing antibody. These are clade B viruses. You can see A, C, D, E, etc. So we had breadth. And then we moved into the first important clinical trial, and I'll just present this and summarize what's new since the last time I was here at the meeting in Paris about a year ago, or more. So these were the various groups of animals that we did the first clinical trial, animal clinical trial on. This is the dose, and this is the QS21 that I mentioned to you before that we no longer can get our hands on. And the important one is comparing group 5, just to come to the bottom line, to group 3. The protocol is shown in this slide, and each one of these is when animals were vaccinated. It's a very long experiment. You can get very old doing vaccine experiments. So then, week 135, 9 more months, sacrificed and examined the tissue for viral sequences. We looked at most tissues of the animal, not all tissues. And the results were shown in this slide in summary form. So if you look at the vaccinated group versus the naive group, you can see that we failed to get sterilizing immunity. In other words, all the animals had the same height of viremia, virtually, as the controls. I mean, no, they didn't, but it's not a big deal. They all had viremia. But the difference is, ultimately, they had no detectable virus in the plasma. Excuse me. More interesting, at least to me, were the results at the bottom. At the very end of the experiment, I told you that we harvested tissue. And if you look at the naive, and you look at various tissues, you see we could detect viral sequences. But in the vaccinated group, only one animal, and only when all the lymph nodes were pooled, did we find any detectable viral sequences. We find that those animals had high antibodies to CD4I epitopes. For those of you not familiar with this, what do I mean by CD4I epitopes or CD4I antibodies? Mean epitopes that are induced by the binding of CD4 to GP120. So when GP120 alone, GP120 bound to CD4, you have new antibodies. Those antibodies are called CD4I antibodies, and the epitopes that they target are called CD4I epitopes. And the reason that you conclude that these are CD4I epitopes is a competition experiment with monoclonal antibodies that are specific for CD4I epitopes that are known in the literature. So the animals that had these high titers were those animals in the vaccinees. They all had reached high titers before challenge of these particular kind of antibodies. And protection correlated with the broad neutralizing antibodies for the co-receptor binding domain of GP120. In other words, with the CD4I antibodies. So we had a correlation, albeit the great limitation of number of animals. So CD4I responses correlate with control of infection among all the envelope immunized macaques. As you can see here, these are those who had titers greater than 200, less than 200. This is the tissue viral load in total. Okay, now what's happened since these experiments? And I will summarize in the final couple of slides. So I would say that these first primate results were interesting, and they suggested an effect of CD4I antibodies in providing protection against a heterologous challenge. We had two heterologous challenges. The number of animals was, however, limited. A second, just completed, well, the slide was made a few months back, so it's not just completed, but a second result was completed in the last year and repeated the results of the first one very closely. A third primate study with repeated low-dose challenges, turns out they weren't so low, over a much longer period of time to keep challenging the animals was far less impressive. The first reason we could think of was that, like all envelope-based immunogens, the humoral response was not sustained. That was actually true. We did not sustain it. So that's the most obvious reason. But there were also other major differences. This third study was with a different adjuvant for reasons that I told you about earlier. And the full-length single chain was expressed in CHO cells, whereas the first and second trials, it was expressed in 293 human cells. Could that be an important glycosylation difference? So, we're doing a fourth trial with the Gates Foundation, a fifth trial with NIH, and one gets the feeling we'll be doing primate trials until hell freezes over. But on the other hand, the Gates group has indicated a willingness to go forward with phase 1 trials as we are. We have very strong reason to believe that a DNA prime, these are all just with protein, but a DNA prime protein boost will give greater sustenance of the antibody response and will give greater titers. We've seen that with other groups, and we're starting to get such results ourselves, so we'll probably be switching to DNA prime protein boost. We're also testing using the vector VSV because of a relationship that we had with Wyeth that turned over their technology to a spin-off company from our institute called Profectus, so that we have the rights with VSV. So it's a natural, and some of the leaders of that program are pushing for that as the priming dose, and those trials are starting to go now. We did not achieve sterilizing immunity, as I already noted, but we obtained protection. The pattern of protection, to me, suggests an ADCC mechanism, that is antibody-dependent cell killing, and that's now under study, and actually the early results indicate that there is potent ADCC by this specific class of antibodies, CD4I antibodies. We're testing various adjuvants for prolonging the antibody response, and before I read the last point, I want to tell you that we have a new technology for rapidly isolating memory B cells from someone's peripheral blood, expanding them, cloning the genes, and developing monoclonal antibodies very rapidly that can look at the archival evidence of what was the immune response of this individual. Sort of gives you an idea of what infections you had before also in some cases. But in the case of HIV, some of the people who live a long time and suppress virus without any therapy have very high titers, not found in their blood, but found in their memory B, that indicate that they were producing very high titers of CD4I neutralizing antibodies and with ADCC activity. Future HIV vaccine efforts should be based on adequate primate results, they haven't in the past, using heterologous challenges before clinical trials, with good rationale for vaccine and transparency in a decision-making process that selects candidates for trials, which has also not always been the case. My coworkers are particularly George Lewis and Tony DeVico at the Institute, Tim Fouts, who's now in the spin-off company called Profectus, and our collaborators are listed there. John Eldridge is the guy from formerly at Wyeth who brings the VSV technology, and John Gershoni, Tel Aviv, Roger Paul at ABL have also been involved. Thanks.